useful shared. So
Brilliant, thank you. So it falls to me to say welcome to you, everyone who has joined us. I understand we haven't quite met the bar for the most registrations for one of our webinars, but we should have a lot of people. And as Vipka says, this will be recorded, so many people can watch this afterwards. Vipka, could you share your slides for my introductions? Here we go. There's our housekeeping slide again. And it's just a reminder, this is going to be interactive at the end of the meeting. So please put any questions for our speakers in the Q&A box. Next slide, please. So welcome, everyone. This is the first of a series of webinars that the FVE is going to deliver on animal health matters. My name is Jane Clark, for those of you who don't know me. I'm one of the FVE board, one of our vice presidents, and I'm also the director of veterinary services in the UK for the Food Standards Agency. I have additional interest here because for a period of time, I was the veterinary lead for avian virology in our UK's National Reference Lab. And what this webinar is going to help to deliver is make the connections between the FVE's pandemic preparedness paper and the work that we know is going on in the EU and globally in terms of preparedness for high-path AI. If we could go to the next slide, please. A little bit about the FVE for those of you who don't know, and we've got a little QR code in the corner. So if you take a photo of that QR code, you will be driven to our website. It's really interesting to see. We have a membership of 39 European countries. We represent over 330 veterinarians across that membership. And as you can see, our mission is around enhancing animal health, animal welfare, public health, and protecting the environment. And this all comes together when we're talking about high-path AI. Again, if we could move on to the next slide, please. Thank you. At our last General Assembly, we had a vote. We asked people what their main animal health concerns were and what they would like to hear about. And as you can see, high-path AI is slap bang in the middle of that word cloud. So that's our first webinar. We've got a webinar coming up in April on blue tongue virus, and then we will also think about some further webinars judging from the level of interest and concern. But in particular, high-path AI is important for, next slide please, Vika, our thinking around pandemic preparedness. It's one of those exotic notifiable diseases that we really are concerned about in terms of human pandemics. And the FVE developed a position paper and adopted it at our General Assembly in November on the contribution of the veterinary profession to pandemic preparedness. And it's at this moment I do want to make a special shout out to our colleague Simon Doherty, who we lost last year, and who was a driving force behind this paper. It's proved very timely. As you can see, we have recent news around the UN pandemic agreement negotiations and so on, and it really gives us a framework to engage internationally on this. Next slide, please. So we identified a number of areas where we believe veterinarians are crucial to pandemic preparedness. And these infographics are available. We're using them on social media and so on. And we think this will be a really helpful way for us to communicate our role. I'm not going to read out all of the slides. As Vika said, there will be, the slides will be available, and you can also actually view the whole paper on our website. But pandemic preparedness does constitute a number of areas. So we have divided those up into early warning systems, disease prevention, monitoring and surveillance. And next slide, please. Innovation, biomedical innovation, One Health collaboration, contingency planning, and of course, and I think hugely important, communication and education. And this is one of the things that we're doing with this webinar. Next slide, please. So we have some recommendations in place. And I was at a meeting yesterday when someone said that the human brain can keep 3 concepts at any one time. So we've tried to divide these things up into 3s. So our recommendations include developing risk-based contingency plans, recognizing that role of the veterinarian, our expertise and our role in society, I think, is really important in terms of pandemic preparedness. And of course, that buzzword, the One Health approach. And to that, I would also add welfare, because if we get the health right, we get the welfare right of people and animals. And I think you could say the welfare of the environment as well. Next slide, please. So again, 3 things. We suggest a One Health way forward. And to keep it simple, that's prevent, respond, and communicate. They don't necessarily go in that order. It's a cycle. And we think communication is a really important aspect to ensure that that public messaging helps to drive cooperation and investment. And I'm not going to spend much more time because I think it's much more important that you hear from our guest speakers. So if we could just go to the next slide, please. You'll see one of the reasons why we really care in terms of pandemic preparedness about avian influenzas. We are seeing a multi-host ecology, and our speakers, Ian and Alessandro, are going to talk to you about it in a lot more detail. So if we could just go to the next slide, which is just a little introduction to our speakers. And if you could just bear with me, I've got to open up my, my background. I hardly need to check Ian's CV because he used to be my boss. So I'm really, really delighted that Ian can join us. He was the head of virology for over 10 years in APHA, that's the Animal and Plant Health Agency in the UK. And then he became the Scientific Services Director. He's the former director of the OIE FAO International Reference Laboratories for avian influenza, Newcastle disease, and swine influenza. And he holds a visiting professorship in avian virology at our University of Nottingham. He's also an honorary professor with the Royal Veterinary College in London. I'll introduce Alessandro Broglia later as he's about to speak. And at this moment, I'm going to ask Ian to be prepared to put his slides up and speak to us about his overview of avian influenza spillover events. Ian, boss, can I hand over to you, please? You're on mute, Ian. It had to happen.
That's the one mistake. Yeah, thanks very much, Jane, for the kind introduction, and good morning, everyone. And it's a pleasure to have the opportunity to kickstart your webinar series. And obviously, your questionnaire flagged HPAI, and why? Well, because there's a huge amount going on. There's been exceptional global spread. I mean, it is a panzootic. There's very high infection pressure. We're seeing domestic birds suffering right across the globe. There's heavy environmental contamination, and that's led to much greater exposure to wild bird populations that perhaps in the past weren't exposed. We've seen spillover to mammals, principally scavengers that feed and predate on sick and dead wild birds. But we have seen evidence now of sustained transmission in mammals. And of course, we can't go far without listening to the dairy cattle story in the US, and that really has changed the landscape in terms of our thinking about how these viruses can jump and maintain. And yes, we've got a virus that's evolved over many years now and has become what we call very highly fit. It is a good fit for being able to jump between hosts. The good news, if there is any, is that antigenically, these current viruses that have spread around the globe are relatively conserved, and that may offer some pathways of hope for vaccination in the future. So you've already seen some slides showing this is the global picture just over 12 months ago, noting probably those spots in Australasia are H7 viruses. They're not H5N1. And that's really what I'm going to talk about today, because that is the virus that's caused the panzootic. This is the situation since October 24. You can see not much different from the previous slide. Perhaps there's a few gaps in there, and you think perhaps Africa's free. It absolutely isn't. These are just surveillance blind spots. And where all the spots are is probably where surveillance at its best. This is the European situation, which you'll all be familiar with, where we had a disease that perhaps only came into Europe every 5, even 10 years, now is a seasonal event. And in fact, even in between events, we've had activity 2 years ago where the virus maintained through the whole year. So we're at a much elevated risk. Why is this happening? Well, there's 2 fundamental things I need to say about the virology. The way the virus changes, and that's the key to why it's doing what it's doing. So you get point mutations in the external glycoprotein. So you've probably heard of H and N, the 2 numbers that give the serotype. Well, point mutations cause what we call antigenic drift, and they can get fixed in the virus and can give the virus an advantage to spread and maintain in a population. The far bigger mechanism, of course, and that gives rise to what we call clades. So you may have heard of this thing called clades, which is highly confusing, even when you work in flu, let alone outside of it. But genotypes are defined by the constellation of genes that the virus has. So each virus has 8 genes, and it can shuffle those genes as it mixes and matches. So 2 flus co-infecting the same host can exchange and shuffle those genes. And because these viruses maintain in wild birds, they have plenty of opportunities to mix and match with other strains. And that is also a clue as to why these viruses are being so vigorous. Now, this is a dendrogram of the H5 virus. And although we've had these viruses around for about 10 years now, this little group at the top, I'm going to talk about this almost exclusively today. It's called the clade 2.3.4.4, and there's a subgroup called B, and it's that group that has been responsible for transglobal spread. And there you have a diagram showing multiple epicenters. So there's not one place where these viruses are spreading from. There's multiple centers where they're now established around the world. And indeed, lots of dogma are getting broken by this virus. The virus made it from east to west across the Atlantic. We never thought that was possible. And that implicated pelagic birds that had not been shown to be associated with the virus before. As a result of this big spread, many populations of birds, and you can just see there, just about every order of bird, we have to consider susceptible. Some are more important than others, but now we're talking an exceptional wide range of species as the virus gets into new environments where it's not been before. Indeed, some of these will be very susceptible. We know, for instance, the good old anseriforms, the ducks and the geese, they are still the critical reservoir. They're still the major transmitters. They are largely asymptomatic, not always, and therefore that enables them to be able to carry the virus from one location to another. Of course, that virus will then spill over into other populations where it may have more profound effects. So we know in Europe, looking at swans is a pretty effective way of doing passive surveillance, simply because they're big and white and people report them. Of course, the virus is still very highly lethal in galliforms. But then there's secondary hosts, these seabirds, where outcomes are not always fatal, but quite often. And then we've got these other hosts, birds that feed around farms. We're not entirely sure what their role is. They certainly probably got a role in mechanical transmission, and they can become directly infected. And then there's other hosts that we observe, despite exposure, they don't appear to get sick. Obviously, a lot of research needed to understand the basis for that. But of course, because of this, we're sitting on a biodiversity disaster. Large populations being severely affected with large-scale mortality events. And indeed, this analysis here showed that you've got 16% of birds and 27% of mammals that are on the red data list, endangered, that have been infected with this virus. So these are really serious impacts. Now, a critical host around the world is the gulls. These are a relatively new feature in the last 5 years. They've always been susceptible to flu, but not H5. But the particular virus we've got now is very good at infecting gulls. And they are perfect bridges from water habitats to areas where we're farming poultry. So they're quite important. Now, as a result, I mentioned already that there's been a spillover to mammals. These are mainly scavenging mammals, and you can see there's a big long list there. It's also got into sea mammals, pinnipeds. What's the importance? Well, they're dead-end hosts in the main. Okay, so the virus doesn't maintain in foxes, it doesn't maintain in mink in the natural environment. That's probably because of their contact structure. That doesn't lend to easy transmission of an infectious agent. But other species, such as the sea mammals, there has been more association. I'll come back to that. You can see here as a correlation. Domestic birds are in dark blue, which of course we monitor very closely. Wild birds, it's more patchy on how well we monitor them. That will vary by country and by region. And then at the bottom, you can see the turquoise. And there's a direct relationship between the number of cases in mammals and the number of cases in wild birds. So the wild bird cases go up. It's logical. There are more of these scavenging mammals becoming positive. And I should say, the disease in those mammals is a really acute nervous disease. The virus reaches the brain very rapidly and death. I mentioned the mammal-to-mammal transmission. So this is the case in South America where they've got huge numbers of sea mammals, as any of you who've been there will know. And the virus, because of those communities that they come together at certain times of the year, that is an environment that's conducive to enable the virus to spread. And indeed, mammal-to-mammal transmission has been proven. Now, why is that important? That's important because this virus is still a bird virus. It wants to be in a bird optimally, but if it gets into a mammal, will it make genetic changes through those mutations I've described that enable it to infect people? And of course, that means, does the One Health risk change because it's in a mammal population? We've already heard Australia, Antarctica, sadly, a couple of years ago, had the first cases, and we are still seeing cases there. Thankfully, it's not gone on to a huge scale, but the fact is the virus is entrenched, and it's hard to see how it's going to dissipate and remove from those populations. Of course, there are other factors that enable the virus to persist. So this is a mink farm in Spain in October 22. These mink are in these open-sided cages. They were exposed to gulls. The virus started transmitting through the mink. And of course, the question then is as it transmits through the mink, does it force it to change genetically in a way that its phenotype changes and it might then infect people? Now, thankfully, this one didn't, and the farm was depopulated and the outbreak controlled. In Finland, there was a higher number of cases, a substantial number of cases in farmed foxes. So these are Arctic foxes, and there was secondary spread. So by the time the authorities realized what was happening, the virus was already transmitting. Of course, that's quite dangerous. If you allow a virus to transmit in a mammalian population that's in close contact, that creates more risk and challenge. There were changes that occurred in the virus, but thankfully, none of them, as an aggregate, actually really changed its risk profile for people. The Finns did a good job. They depopulated these farms, and they took the decision to offer prophylactic vaccination to the fur farm workers with H5. I think the uptake's not been good, but it has been offered. So it shows about the One Health thinking. Now, what's this diagram? At the top, these swathes of color represent different genotypes just in Europe. Okay, so I'm only looking at Europe here. And you can see there are lots of genotypes emerging along the bottom there, but you can see that there are certain ones that seem to dominate. This is the number of cases, okay? And this is the natural cycle of selection that we get with the virus. So huge number of genotypes being described, but only a few of them are probably really important and relevant in terms of the long-term epidemiology. Now, of course, as the virus gets into other continents, it mixes and matches with local strains there. So the American strains look quite different to the European strains. So that cascades another set of reassortments. All these colored bars show genetic diversity. So what it's telling us is, even though the H5 part of the virus, the hemagglutinin, is quite stable, the rest of the genes are changing all over the place. And that creates a lot of diversity and a lot of risk. So it's the virus that's changing here. And I would say that I've been working with these viruses for a long time now. This particular strain is the most fit and infectious of all H5s. So that leads me into the dairy cattle. We've got a situation in the US that started March last year. We've got nearly 1,000 cases now in 17 states. Well, what is happening? We didn't see this one coming. It is an infection of the mammary gland. It's not a respiratory pathogen. We've had several spillovers to date. I'm hearing that there's possibly another one now. That's a concern. Multiple spillover events. For most of the time, it was only a single spillover, but recently we've heard of more. You get a reduced feed intake, you get a milk drop, a substantial milk drop, mastitis, but symptoms can be quite variable. but there's an extremely high quantity of virus in the milk. And that is a real concern. How's it spreading? Well, it looks like it's by a movement of infected cattle. And those of you who are familiar with the dairy industry in the US know they move their dairy cattle an awful lot. I've certainly learned a lot about that. And then the fomite spread as well. Complex pathway for wild birds. Ultimately, that's where the virus came from. How did it find its way in? We really don't know. But it's certainly now maintaining efficiently in dairy cattle, not beef cattle, because it's only an infection of the mammary gland. Now, consequent with all that exposure and spillback, I might add, to poultry, there's been an increased awareness and monitoring of humans in contact with dairy cattle and poultry. And you can see here, we've got about 70 cases now. Now, thankfully, only one of those has been severe with a fatal outcome. Most of them are mild conjunctivitis, but a few of them, depending on underlying health factors, can be a bit more invasive. Pasteurization does kill the virus in milk. But of course, in some communities, drinking of raw milk is still a practice that's followed. So what's this? Well, this is telling us we can track all of these viruses. Whoops, oh dear, what happened there? Right. We can track all these viruses and we can understand, can they bind into the upper airway of humans? So if it's going to be a zoonotic virus that's going to successfully transmit one person to another, it's got to bind in the upper airway. So the red line is the human airway and the blue line is the bird airway. And this is a virus that did infect both humans and people here. And you can see that it actually binds both. But if you look at all of these other viruses, these are the European ones. They only bind the human, the avian airway, not the human. And likewise, the cattle viruses. And we've been tracking all of these mutants as they emerge and evolve. And they solidly want to bind in the avian airway and not the human airway. Now, that's good news, but we need to be watchful in case it changes. Yes, this virus is spilling back into domestic poultry. The dairy cattle have got more limited control over them, movement restrictions and testing between states, and they're now running out on bulk milk testing at state level and then tracing back to infected farms. But the measures are not as aggressive, and therefore you've seen the level of spread. And of course, that does create problems because the virus is getting back into poultry that might be adjacent to an infected dairy farm. And of course, there are very strict measures for control of the disease in poultry, culling of the flock, etc., movement restrictions, disinfection. So the consequences of the cattle infection are having an impact on the poultry industry. Now, of course, other species can inadvertently get exposed to the virus. Domestic and companion animals have been a question for a long time. Yes, the virus has been getting into cats. And again, as with the scavenging predators, you get an acute neurological disease and you get, invariably, you'll get rapid death. These cats, how are they being exposed? Multiple pathways. Cats in milking parlors, drinking obviously milk, exposed to high loads of virus. Raw pet food that's been untreated, that's sourced from birds that are potentially infected or contaminated with the virus. There's been big outbreaks in zoos that have had an impact. And there was a case in Poland 2 years ago where a very large number, in excess of 30 cats, were all independently affected at the same time. The conclusion in the end had to be that there was a common food source because these were house cats and they were right across the country, rather than lateral transmission between cats. Dogs, a bit less clear. In theory, they are susceptible, but there's much less report. But we have to consider they're a potential host. And of course, that brings a more magnified risk in terms of people contact if the virus is in companion animals. Can vaccination solve the problem? Well, of course, the answer is it can, but it can't be the only tool. And I always put this slide up when I talk to industry. It's not a substitute for weak biosecurity. So there has been a big paradigm shift in thinking around vaccination. It's been a taboo for Western countries and exporting countries because they want to protect their rural economy. But I think that the tide is changing, and we've had a vaccination program in France, as you will be aware of, in their duck sector. Other countries are looking to follow suit. There's even a hot debate in the US now about vaccinating layers and turkeys. So things are changing. And I think probably if we roll forward 5 years, probably we'll wonder why we were having a debate about vaccination. Clearly, there are a lot of benefits. There are some challenges. We need to be able to monitor vaccinated populations and track for the incursion of wild type virus. And Europe at the moment is trying to refine how it does that surveillance in an effective but cost-effective manner, because the cost of the surveillance that was initially set down was very cost prohibitive and unaffordable to industry. Of course, it has the benefit that it lowers the human exposure risk, reduces environmental contamination, which reduces risk of onward transmission to wild birds. And of course, in many countries, this has a big impact on protecting food security. So to wrap up, we've got exceptional global spread. We've got a panzootic that's infecting most continents. And frankly, I think it's only a matter of time before it finds its way into Australasia, sadly. It is a big threat to biodiversity. The dynamic of changing wild birds has really changed the landscape. And of course, we will continue to get an oscillation in terms of the number of domestic bird cases. And of course, in Europe this winter, we've had more cases than the previous winter. So it's not a case that we're in a declining scenario. We've got the mammal dimension. And of course, what is going to happen with the dairy cattle outbreak in the US? It has not stopped. If anything, it is being maintained and continuing. And I know that the Americans are looking seriously at vaccination in cattle, and field trials are underway looking at vaccine. It could be a bizarre situation. They vaccinate their cattle before their poultry. And of course, we've got this virus is continuing to change. It's not standing still. So we have to be on top of that. But there are options for vaccination that are realistic, and there are vaccines out there that can do the job. So I'm not going to go through all these people. A lot of the data I've shown is drawn from various consortia across Europe, internationally, and in the UK. Thanks very much.
Wow, thank you, Ian. Can I just also say spot on 18 minutes, very impressive. That was a lot of information for you all to take in. As I say, there is a question and answer box. You'll find the link at the bottom of your screen. There are no questions yet, but please do put any questions that you have for Ian into that Q&A box, and we will deal with that at the end of this session. I'm now really, really delighted if Alessandro can put his camera on and get ready to share his slides to introduce Dr. Alessandro Broglia. He's worked for EFSA for 15 years and is currently in the Unit for Biological Hazards and Animal Health and Welfare. As a senior scientific officer, he's tasked with risk assessment relating to animal health in particular, transboundary diseases, analysis of emerging drivers in agricultural and livestock systems, mapping of livestock-related patterns, scientific support to the EFSA panel on animal health and welfare and its working groups, which is a lot to do in your day job. So we're very grateful, Alessandro, for you coming to speak to us about the zoonotic avian influenzas and enhanced preparedness. And if I could ask you to share your screens and tell us all about that. Thank you very much.
Thank you, Jane. Yes, pleasure for me. I hope you can see the presentation mode. Yeah. So
yes, all good.
Good. So thank you for the nice introduction, and also for the invitation to this webinar, which comes really timely because yeah, we have been busy for sure, as you know, with EFSA in the last many years with the avian influenza, and more and more we are collaborating also with our we call it sister agency, the European Center for Disease Control, so under a perspective of One Health. And the topic today will be zoonotic avian influenza and enhanced preparedness. This is the result of a scientific opinion published at the end of January, as I said, together with ECDC colleagues and also with the European Reference Lab for Avian Influenza. I'm a veterinary epidemiologist. I'm not a pure virologist and a top expert on avian influenza, as Jan instead is. So our remit is pretty broad. We deal with a lot of projects and animal disease. And I hope to be able to answer all your questions. I will speak also about public health aspect on behalf of ECDC. Then let's have a nice discussion afterwards. As I said, this is the scientific opinion recently published. That's the link where you can find it. A lot of people involved. For sure, the panel on animal health and welfare from EFSA, the ECDC colleagues, ECDC and EFSA external experts, the colleague from the URL, really a multi-weapon job and the One Health experience, where I also myself learn a lot about different aspects of avian influenza virus. So I'm delighted to present it today. So good that Ian already discussed about the full epidemiological situation in birds, in wild birds, domestic birds, dairy cattle. So I will be very, very brief there and maybe just give a couple of hints about the human cases. Then I will show you what we did for the mutation analysis and the phenotypic traits. And then some recommendation about surveillance, both in animal and public health, as well as prevention and control measures. So here, very fast, the situation in mammals in 2024. You see that basically the major contribution is from Northern America, I would say US, where you have different categories of animals infected. We heard from Ian the situation in dairy cattle, but also some goats, alpaca, and pig, although those were from different genotypes. We had some peridomestic mammals, some mouse species, the cat, and a lot of different species of wild mammals, mostly carnivores, both from the wild or from zoos. Canada, also we see some species there, although much less cases. And Vietnam, in safari zoos, large felines infected, and the Antarctic region with semi-aquatic animals like the seals. In Europe, since spring 2024, we don't have mammal cases. We had before, again here with carnivore, wild carnivores like red foxes, raccoons, otters, and really case of spillover. Of notice, the H5N5 cases in Norway and in the Netherlands, in foxes and the pine marten. But I would say that the situation in mammals in Europe has really decreased, or no cases since spring last year. So for the human case, this is the kind of historical situation from 97 up to now of avian influenza cases of different subtypes in humans. You see that the three most contributors to this are the H7N9 subtype. You have it in a relatively small time window from 2013 to 2019, but with quite a lot of cases. That was mainly in China. Then for sure the H5N1 throughout the entire period with pretty high mortality and with over 900 cases. And the H9N2, low pathogenic, also throughout the entire period with 142 cases. And these basically three subtypes covered most cases. In terms of geographical distribution, we see that in the past decades, mostly we had, this is only for H5N1, we had cases mostly in few countries, Egypt, Vietnam, China, Indonesia as most contributors, while in the last 3 years, Cambodia, US, and China, you see on the right side of the graph, are the countries where most human cases were found. I have to stress that the case in Spain and UK are then inconclusive and they were due to environmental contamination, so not real infections. And we had no case of avian influenza in EU. So in terms of the analysis of the mutation that we did towards the zoonotic adaptation of the avian influenza virus, so we have to remember that there are some distinctive characteristics between the avian host and the human host, in particular about temperature and pH of the environment where the virus infects the cells. In particular, in the upper respiratory tract of humans, we have lower pH and also lower temperature. And this is something not really well and established condition for the avian virus to replicate. So some change should occur. We consider most relevant phenotypic traits that are required for this adaptation. So you know that a virus should infect the cell, should be then the genetic material should enter the cell nucleus and then replicates to produce viral protein, but also to replicate the material itself and then finally be released. We identified 5 of these steps, 5 of these phenotypic traits, in particular the one related to the receptor binding, then the one related to the hemagglutinin instability, the evasion of the innate immunity, this replication, so where the viral polymerase plays a role, and then finally the release of the viral particles. The most relevant characteristic where we found most mutations are the ones related to the specificity of the polymerase and the one related to the evasion of innate immunity. So what we did basically is we identified all the mutations that were linked to these phenotypic traits, connected also to the viral subtypes. We attribute a kind of score of evidence to this mutation according to different criteria and also thanks to some experts that help in the shortlisting. And then we retrieve the sequences from the last 24 years and we check where those mutations compare, in which strains. You found all the details in that link. In particular, we found that we have 34 mutations, most relevant ones associated with the 5 traits that are needed for the zoonotic shift. In the end, over the 27,000 sequences screened, very few ones contain all the 5 traits, and they were all belonging to H5 and H9 and 2 subtype, mostly from Asia and Africa. In the EU, the H5 2344B clade had the highest number of traits, but probably due to this to its widespread. And for sure, we could conclude that these multiple phenotypic traits that are all needed for the virus to become epidemic in humans, and then not only to guarantee the infection of the human cell, but also to then infect other cell and be able to do the human-to-human transmission, is pretty uncommon and also a gradual process. In this graph, well, we can skip it for the sake of time, just showing that we have very little with all the 5 traits, while a little bit more with the 3 and 4 traits. These are the number of sequences screened. More important recommendation about this analysis. So for sure, the quickly characterization of the emerging viruses, so the importance of bioinformatics here and the bioinformatic tools. We realized that a lot of experimental studies are difficult to be compared, so more harmonization would be really needed. For sure, as Jan said, 8 genome segments, the mutation can occur all over the genome, so the whole genome sequences is the approach to check this, together with metadata collection, so not only sequence data, but also the other epidata connected to that. The list of mutations that we have described for sure is not something carved on stone, but is changed. The virus will constantly create new mutations, so should be updated. And the importance of a kind of framework that goes behind of these individual mutations. We know that there is also genetic reassortment where multiple virus infection. So it's not only about individual mutation, but really it's a complex system which requires a complex approach. Considering surveillance, apart from some general recommendation that, again, it's about early detection and focus on the subtypes with the high number of mammalian traits, focus also on low pathogenic virus, because we have seen that some of those strains of low pathogenic entail also most of these adaptive traits, and integrate virological, epidemiological, clinical, and human information. So again, it's not only looking at the sequences that you get the full picture, but if you also have a complete situation about different data. Considering animal health surveillance, here the objective in this remit is detecting all those mutations that have mammal adaptation or even zoonotic adaptation. And the approach we consider is mostly kind of risk-based and passive surveillance, targeting at least 3 categories of animals: those with known exposure to infected animals, those found dead in high-risk areas or period, and those showing unexplained signs, again, in areas at risk. Sick or found dead animals should be sampled, whole genome sequence from the tissues, from the target organs, but also repeating the sampling if no culling is applied, because mutations can occur even inside the same epidemic. And here, the category of animals that we may have different surveillance scenarios. So the farm for animals, you heard about the farm mink, for sure those exposed to found dead wild birds, domestic ruminants, but also pigs. We know they can get infected. The risk is especially at mixed farming situations. Wildlife, for sure those found dead, but also rescue centers for wildlife are a spot where the surveillance can be triggered, because there typically sick animals are collected, and also you have different species in the same place. Companion animals, so the cats, for example, the issue of neurologic and respiratory signs in high-risk period areas. And finally, also, we consider rodents, where you have unexplained mortality, especially in the farm environment, if they are found dead, so they may also be a target of surveillance. In terms of prevention and control measures, at farm level, we consider biosecurity is probably the most important preventive measure to apply, because this limits the introduction and the contact between wild birds, the first carrier of virus, to the animals. Contingency plans, even Jane at the beginning has shown that as one of the components for the prevention. We have a lot of species, so continuously new species are detected, so contingency plans are really important in order to get prepared. We heard about the contamination in the environment, a lot of virus in the environment, also due to the infections and outbreaks in domestic poultry farms. So even the farm location and the type of breeding system can be a risk factor, because we know in Europe, for example, we have areas with a lot of farms that are very dense and with a lot of animals together. And there, for sure, we have a lot of biological mass of virus when we have outbreaks in these places. So the structure also of the breeding system should be probably carefully considered and rethought. Yes, control measures, typical ones: isolation, cooling, disposal of carcass. What is important is really to be prompt and fast in applying those control measures. We have seen how for example, the epidemic in dairy cattle in the US has gone fire and really fast on spread, probably also because of the slow application of some movement containment. And this is taught really a very important lesson. For the general public, I would say the clear communication. So raw dairy products from infected areas in dairy cattle are an issue, so they avoid the consumption of those. The aspect of managing companion animals, so for example, their access outdoor when we have outbreak areas. And for sure, for people with backyard farming hobbies, even there could be a place where you have a risk, so hygiene and protective measures should be taken even at that level. For wildlife managers, hunters, rangers, researchers in the wildlife sectors, biosecurity again in handling the carcasses or the animals, waste disposal, and again strengthen the stakeholders network, because wildlife is typically something that is dealt by many actors that sometimes they don't speak to each other very often or clearly or completely, so it's important there to have that. For public health surveillance, I would say to target the symptomatic but also the asymptomatic exposed people according to the exposure. The surveillance in hospital or in healthcare facilities, very important, so in a matter of raising awareness about workers there and clinicians. In the context of areas where avian flu outbreaks are detected, then also the surveillance in the healthcare facilities in those areas is really key. Second aspects are that for sure the positive sample should be sequenced and shared also with the WHO collaborating center. In case of inconclusive samples, then again refer to the national reference lab for the subtyping. Metadata, as I discussed previously, so the information about epidemiological sources and data connected to those samples, the timely notification from member states, and for sure the One Health approach, mostly between veterinary and public health sectors and also across border collaboration. Last but not least, some measures for prevention and control under the public health aspect. So for sure at workplace, measures should be taken in order to prevent the spread. At poultry farm, for example, we have the OSHA, the Occupational Health and Safety Agencies, which clearly highlighted guidelines about that. The preparedness to respond to the confirmed cases, which for sure include self-isolation and contact tracing. And then again, as Jan talked about vaccination, even here for public health and people, the seasonal influenza vaccination, because of limiting the possibility of a reassortment of virus, while the vaccination with H5 vaccine for the person exposed, I think mostly here about farm workers, should be considered a kind of complementary measure. Not yet there is sufficient evidence to recommend this everywhere. Indeed, last but not least is the awareness about the zoonotic risk of avian influenza, especially in the group at risk, so again at farm level, but also those with companion animals or with backyard poultry. So all these situations may be targeting raising awareness campaigns. I acknowledge again colleagues from URL on avian flu and from the IZS from Abruzzo and Molise, who also helped with the mutation analysis. For sure, OSHA colleague, ECDC, Epidemic Intelligence, and also some other external colleagues that helps with the revision and with the collection of data. And I really thank you for your attention. You find the EFSA topic page on avian flu at that link where everything about avian influenza is published. We have now a project running about HPAI in dairy cattle in US. Assessing those situations will be ready in May with the second output ready at the end of the year. So I encourage you to stay tuned and check those. Thank you.
Thank you very much, Alessandro. Stay tuned, I think, is a very important message. And Ian has come back on screen. We had a number of questions posed in the Q&A. And in fact, if Alessandro, you want to have a look at the Q&A and say whether there are any of those questions that you would like to answer. Ian has already said that he would like to answer Bert's question, which was around vaccination, but what species, not only birds, but other species to prevent infection, or are there, because of organ system differences, challenges for vaccination in those other species? So Ian, over to you. And Alessandro, you have a look at the other questions.
I'm happy to answer any of those, Jane, but I'll leave you to moderate. But a good question. We've got to be clear, before we go off and start vaccinating all sorts of animal populations, we need to understand what the risk pathway is, how the virus spreads, what it can maintain. Clearly, there is a big debate about cattle, and the fact that the Americans have already commissioned clinical trials some months ago now shows their thinking. You're absolutely right. How can we vaccinate dairy cattle and assure that you're going to get protection in the mammary gland? And you know, your vets, you all know better than I, but the fact is it's an immunologically privileged site, so this may be a challenge to secure that. So I think we need to look at contingency. So part of contingency preparedness should be, do we need to use prophylactic vaccination? What if this virus was to spread to pigs and transmit between pigs? You know, we need to think about what we're going to do, because the concept that we're going to go and cull tens of thousands and millions of pigs like we do poultry may not be acceptable or actually indeed effective. So we do need to look at contingency, and I know there is some interest in taking that forward in Europe at the moment.
Thank you, Ian. And I think that leads very nicely on to another question, which is the panel's view. So please choose between you who would like to respond, perhaps both of you, on the US policy change to stop depopulation of poultry houses affected with avian influenza, the relaxation of rules for people who keep backyard flock poultry, and what do you see of the risks and, if any, and advantages of these changes? Who would like to start?
Well, I'm happy to go.
Go on, Ian.
Alessandro, I'll go first, and Alessandro. So, in brief, we haven't seen the detail, but my understanding is they're only talking about partial depopulation. Now, we know from our experiences in Europe, once you get an outbreak on a site, biosecurity protocols and activities go out the window. So, protecting the rest of your site and the internal perimeter is extremely challenging. Now, they talk about treatment. I can only assume treatment means that they've been thinking about emergency vaccination, which we know on a farm that's already infected will be a non-starter because of the time it takes to build immunity. So I think we do need to see the detail. But let's be honest, people have been grappling with the effective control strategy for years, and I would be very unconvinced about the idea of leaving susceptible birds on an infected site. That's first thing. The backyard question. Well, I mean, yes, we have to be careful. Backyard don't necessarily lead to the introduction of virus to the commercial sector. I always refer to them as sentinels for wild birds. What you've got to consider is there's an increased public health exposure when you've got backyard birds. And the lady that died in Nevada kept backyard birds, and it was unequivocally shown that the virus she acquired was in her backyard birds. Now, it's true that she had underlying health causes and stuff, but you've got that dimension as well.
Thanks, Ian. Alessandro, would you like to add anything?
No, that's yes, the importance of backyard. I want to stress that, but Jan already indicated that. And as I said, it's a matter of public health risk. So backyard farming is an issue, and definitely surveillance of that. Yeah.
Okay. I know that you wouldn't like to answer a question, Alessandro, you've said so, but could I just actually, while we're talking about that sort of human companion animal bond and close proximity, ask the question about what do you think the current risk for companion animals is in Europe?
Yeah.
Alessandro, yeah.
Yes, I mean, we know that some companion animal species are susceptible, and there are certain species that could be at risk. So we heard about cats. So we know that cats are not always kept indoor, but have access outdoor. So in Europe, where you have areas at risk, where you have circulation of virus, where you have areas with outbreak in poultry or in wild birds, on those spot situation, I would say that a risk is there for our cats. So in certain period, maybe considering to keep them indoor or to limit that they roam outside is a measure that could prevent some issue. Also because then the infection of a companion animals means a very close exposure also to human in the household. So I think that without generalizing that we have to keep all our cats now closed inside, that's not the point. But in areas where you have active outbreak, very close, then I would say that could be a measure, at least temporarily, to be kept. So also the aspect of feeding, we have this at least suspicion that feeding raw poultry was one of the vehicles for transmission. That is also again another aspect. So maybe feeding poultry to cats again in areas where you have active outbreaks or epidemics is something that should be avoided.
Thank you, Alessandro. Yes, did you want to add anything, Ian?
No, I think there's questions there about cattle surveillance. So I suggest
we'll come on. Yeah, let's come on to that. Absolutely. So the first one, Alessandro, you would like to answer the question from Julie. In an area where H5N1 is circulating and found in dead birds, is the advice to test cows with mastitis for avian influenza?
Yeah. So in that case, I would say that, I mean, we have suggested to consider the inconclusive diagnosis. So for sure, a mastitis where you have a clear diagnosis that is not linked to avian influenza probably you don't need to couple there the sampling for avian flu. Of course, if it is in an area, or even worse, in a mixed farming with poultry or with found-dead wild birds that have been tested positive of avian flu, okay, in that situation, maybe yes. Cows with mastitis, I would really trigger a testing also for avian influenza. So it's a kind of risk-based approach, and where other clear diagnoses are excluded, then I would go for testing cows.
Thank you. That's really helpful. And Ian, there is a question that is linked. Are the WHO recommending screening of dairy cattle? Do you expect the same development of spreading in Europe as in the US? Big question.
Yeah, good question. Well, first of all, WHO don't really comment on veterinary matters. They like assurance that everything is being done, but they've not made a statement to my knowledge. I think we've got to remember that these viruses that are causing the problem in the US are a different genotype to the ones we have in Europe. But that said, there has been work done in Europe that has shown that the European strains that we have at the moment, if they get directly into the mammary gland of cows, they will cause the same syndrome. Okay, so there is a risk. We have had an excessively large outbreak, of course, in Europe over the last 2 to 3 years. So I think the spreading of the virus into cattle, we are aware of it now, and we are looking, and I know many European countries have done some bulk milk testing, they've done some serological screening of dairy herds, they've looked at trends in mastitis cases, and there's no evidence, generally no evidence. So I think you need proportionate surveillance, you need awareness. As Alessandro said, there needs to be a decision tree. I think the notion that we're going to get high path flu spreading in our dairy cattle imminently is not likely, but we do need to be aware that if those risk pathways occur in the US, could they occur here? And it's difficult because we don't exactly know what they are. But I don't think we've got evidence, despite heavy contamination of the environment and domestic birds over the last 3 years, that we have got a problem in Europe at the moment. But we need to obviously make sure we've got the capability to rapidly monitor.
Okay, great. Thank you. And I'm just going to, there's 2 questions that are quite similar, and I'm going to ask for a quick yes, no, before I move to Alessandro and one of the questions he'd like to answer. Should we be testing raw milk production, and should we be testing bulk milk?
Well, we shouldn't be testing raw milk in Europe, in my opinion, because we need to have a trigger to do that.
Yep.
The bulk milk, again, I think a decision tree is needed. I personally think a baseline check, which is what many countries have done, is good. I don't think there's an ongoing need unless you start getting some triggers, and we need to develop a decision tree with triggers in to do the bulk milk testing.
Brilliant. Yeah. And Alessandro, yes, also, no?
Yeah, to the bulk milk also, since it's a point that also the European Commission is pointing attention, because of course we would like a surveillance system that can screen easily and very cheaply everything, all the dairy cattle, and bulk milk seem promising, but there are also some disadvantages. So first, cows with mastitis or those infected, they don't go then to the bulk milk.
They should not.
Also, you have this dilution effect that you have
yes.
Just a little bit of virus, and then it's diluted in the whole bulk milk. Then probably you don't have yet a test able to spot that. So it's a bit tricky, and should be seen carefully.
So I'm going to need to wrap this up with a final question, and I think it is the $10 million question. It comes from John. Alessandro, I know you would like to answer it. I'd also like Ian's views as well. And if any other questions are written down, we'll try and get some answers to you as well. But in Denmark, there was an article in one of the major newspapers recently claiming that there is a major risk of having a pandemic with flu based on H5N1 within the next 10 years. Is this what you see as well, Alessandro?
Well, yes. Yeah, I think it will be in the next 11 years. No, yes, it's a $10 million question for sure. I know that a lot of media are raising this because we have lived the pandemic, so the fear of a new pandemic is always behind the corner. It's difficult to predict, no crystal ball available. What we have seen, at least in our retrospective analysis, looking 24 years back about the behavior of the virus, is that this full adaptation to humans, making able the avian flu virus to spread from human to human and create really a human epidemic or human pandemic, is something uncommon, but I cannot bet on that because we know that the virus mutate continuously, and what we have seen in the past may not necessarily be what we have seen, what we will see tomorrow. So it has the cards to create a pandemic, but we are reassured by this retrospective analysis, and also I can say that we are more aware, and also we have, at least in Europe, I would say that the surveillance system in place is pretty good. It's not fully okay. There are a lot of aspects, especially talking about public and animal health connections, and sharing of information, of surveillance information is still not ideal. But I think that's the key. So to improve the surveillance in order to be able to spot and stop the spread before it becomes really at high scale.
Thank you, Alessandro. Ian.
Right. In short, if you'd asked me 3 years ago, I would have said probably no. But I think what we've been seeing in the last 2 years, we really need to be cautious about. We didn't foresee this virus could get into dairy cattle. We didn't foresee these. We know the virus is changing, so we have to be, I agree with Alessandro, we absolutely have to be watchful. But one thing to remember is we can tweak our surveillance in Europe. We can tweak our surveillance in the US. Look at the interface around the world, where veterinary infrastructure, public health infrastructure is much weaker. So you've got a very large interface around the globe. So that global connectivity and information sharing is going to be critical to understanding that risk. It is still a bird virus. And I don't believe that it's been likened to a flight of steps. And it needs to get all the way up to the top before it can transmit between humans. And as Alessandro showed you, all these genetic correlates, the virus at the moment is probably still only on the 2nd or 3rd step on that flight. It's got a lot of steps to jump before it can come human to human. So that's not being complacent. We need contingency. We need to take this seriously. We need to track what is happening very closely. But I think we have to avoid, fanciful ideas about going out and vaccinating the entire global population with H5.
Yeah.
Totally agree. Yes. Yeah.
I love agreement at the end, and no one asks for a crystal ball, but that's such a helpful context. Could I just say a huge thank you to our speakers, Ian and Alessandro, to Vibke and the FVE team who've set this up, and also to all of our attendees. As I say, this will be recorded, so please pass the message on to colleagues. Thank you for joining us. Thank you for some great questions and a great panel discussion. On that note, let's close the webinar, and thank you very much. See you next time.
Thank you. Bye.
Bye.